All right, we'll go ahead and get started. Good morning, good afternoon, and good evening, everyone. Thank you for joining us for our October of 2020 Global One Health Initiative webinar supported by the NIH Fogarty International Center. Our theme for this webinar is bioinformatics, and we will be hearing from Acadius Lunayo from the University of Nairobi, Kenya, presenting exploitation of translational bioinformatics in animal health in sub-Saharan Africa, and Dr. Daniel Janies from the University of North Carolina, Charlotte, USA, presenting understanding the origins of and countermeasures to SARS coronavirus 2 and other human coronaviruses. And Dr. George Obiero will also from the University of Nairobi, Kenya, will be joining us for the panel discussion as well. And our moderator will be our own Dr. Zelalem Mekuria, an expert in microbial genomics, who also serves as Associate Director for the Infectious Disease Molecular Epidemiology Laboratory, or IDMEL, here at The Ohio State University. And Dr. Zelalem will provide a more detailed introduction to our speakers at the beginning of the panel discussion. And with that, I will hand it over to you, Acadius, and you can go ahead and share your screen whenever you're ready.
Thank you very much.
Okay, thank you everyone. Let me introduce our today's guests in the meantime. Thank you, Dr. Binkley, for giving me this opportunity to moderate the today's session. As said, we have 3 guests today, 2 from University of Nairobi and 1 from University of North Carolina. The first speaker would be Dr. Acadius Lunayo from University of Nairobi. He is a research fellow at the Center for Biotechnology and Bioinformatics at the University of Nairobi. And he conducts research on regulatory RNAs. He's interested in post-transcriptional, structural, functional relationship of these regulatory RNAs. He's interested in combining experimental and computational approaches to explain cellular physiological functions and the role of regulatory RNAs. With him, also from University of Nairobi, we have Dr. George Obiero, which he will be following and presenting in the first section. I will go and introduce Dr. George Obiero as well. Dr. George Obiero is a director of the Center for Biotechnology and Bioinformatics at the University of Nairobi. And he's also a senior lecturer at the College of Biological and Physiological Science at the University of Nairobi. Dr. Obiero is an established biotechnologist, bioinformatician with extensive experience in academic and industry settings. He is currently a board member of the Kenyan AIDS Vaccine Institutes, and he's an expert reviewer for a National Biosafety Authority. He currently supervises 3 PhD students and several master's students. So Dr. Obiero and Mr. Acadius Lunayo will present today on a title, Exploring and Utilizing Translational Bioinformatics in Animal Health in Sub-Saharan African Countries. The floor is yours.
Thank you very much, Dr. Zelalem. Today, from the university, we'll be presenting on exploring and maximizing the utility of translational bioinformatics in animal health in Sub-Saharan Africa. But first of all, I wish to pass the apologies of Dr. George Obiero. He is currently a little bit engaged. I am under his mentorship on under a program, Future Scientific Leaders and Managers. So he's given me a go-ahead to proceed, and then he will join us later on, if it will be possible. So as a quick introduction, as Zelalem was given, I will not add more, but I'll proceed with the presentation. So generally, the presentation is generally targeting at how do we use translation bioinformatics in sub-Saharan Africa, and with a specific focus at the center. And with that said, I will quickly give a small view of exactly what the center does. Generally, as already said, I'll just take a quick moment to pass a message of who we are, what we do, and our collaborations. And generally, we are located at the University of Nairobi, and we do a wide variety of thematic areas ranging from health informatics, agriculture informatics, industrial technology, and bioinformatics application in all the areas that are listed. And with these, we actually literally attract research from all over sub-Saharan Africa, as we shall see later on, specifically focusing on those particular areas. And we have quite a number of areas specifically focused on translational bioinformatics in sub-Saharan Africa in animal health. We have a number of collaboration across the world, including the one that we are currently carrying out with the webinar, the Ohio State University, and in different places across the world. And we intend to increase as much as we can so that we can have a bigger impact generally in sub-Saharan Africa and possibly the rest of the world. With that said, I'll quickly move to today's topic on how do we use data-driven animal healthcare in sub-Saharan Africa to actually ensure that the research we actually do in the labs actually reach out to the intended users, the end users. So generally, the topic will be from the genome to the phenotype, the data-driven healthcare of animal healthcare in sub-Saharan Africa. Generally, we know that there is a new era of data-driven approaches in health. We have a lot of advanced high-throughput technologies that are rapidly producing a lot of data. This data is volumes of data and include data from DNA, RNA to proteins. And for us to actually make sense of this particular data, we have to connect this genetic information to cellular function and biological behaviors. And this requires us to develop very good approaches at higher levels. And this is very important because for this to actually be effective, these approaches must target how are we moving this data that we're having into an actual product that can be used on the ground. Generally, at these higher system levels, most of the time we want to understand how do we move from a healthy state of physiology to a disease setting. And with this in mind, we need to actually analyze large data sets. We need to analyze and monitor large data sets, either from the biomedical field, healthcare and wellness, so that we can move it from individuals and convert this data at a population scale. That is when the impact will actually be felt. And fortunate enough, bioinformatics does a very good job of actually optimizing this data, this translation data, by transforming the data into diagnostics, prognostics, and therapeutics. We have drugs, assays, and all these different types of products that we can have. And therefore, when we combine these numerous aspects of clinical science and combine them with bioinformatics, incorporate them with statistical genetics and data mining approaches, we refer to that as translational bioinformatics. And as you can look at the diagram that is on the screen, you notice that we have different forms of omics that exist in bioinformatics. And these particular different omics can be integrated through data mining or whatever method that we can have to create an hypothesis. We confirm this hypothesis, integrate some clinical aspects, and then we have very targeted approaches. We are moving from a general approach to a particularly targeted approach. Just to put it as shortest form is in translation bioinformatics, we develop methods that store, interpret, and analyze very large genomic data sets to optimize the transformation of these data into proactive, predictive, and preventive and participatory health. Looking at the diagram that is indicated here, we have a bench side, and then we have the bedside. And we have a barrier from the bench side to the bedside. For us to bridge this barrier, we need to store the data we're getting from this particular informatics, that is data. As we translate the data from this particular data to knowledge by storage, interpretation, analysis, we need to translate this particular data into meaningful insights. In short, saying that we have to move the molecules into the population. And this is one of the biggest drawback that we have in bioinformatics. But we're trying the best we can so that we can store this data interpret the data that we're having, analyze it, and make it available at the at clinical levels. So generally, that is what translational informatics, bioinformatics entails. But why is it important? Why should we really bother about this translation bioinformatics? In this case, these methods that we've built or developed during translation informatics can be used to improve predictions, especially if we have, say, a case of a population scale prediction required, can be used for early diagnostics. You can monitor severity and effects of therapeutics, and as well as in prognosis of diseases. This is a very important aspect, especially if you're talking about life sciences, because every single aspect of this is actually very useful in development of a method that can be useful in either disease treatment, prediction, or any of those. Furthermore, understanding of the molecular basis of diseases by integrating the areas of bioinformatics, clinical informatics, statistical genetics, and general informatics. That means general informatics involving machine learning, data mining, any of those. It will actually be a very important aspect because this will aid us to understand how do diseases actually arise, or how do they propagate from one point to another, or how do they move from one cell to another in the body. This can only be done if you're doing proper predictive methods, because traditional molecular biology can actually pinpoint where they are, but how do they spread, how do they work? It's an interesting field that we yet to venture much into. And finally, by analyzing the voluminous amounts of data, this particular data given, whether it's molecular, clinical, we actually obtain very valuable clinical information, which can be readily applied in any particular disease setting. So the question we ask ourselves, why will translation informatics be important in sub-Saharan Africa? That is a very important question. That is specifically a question we ask every single day at the Center for Biotechnology and Bioinformatics. And our basic answer is animal production in sub-Saharan Africa has a massive social and economic footprint. It is every single country at least has some element of the economy that has animal production. And so if we apply these novel methods, we actually bridge the gap between bench research and real world application to animal health. And in simple terms, putting that we are actually providing products on the ground that can actually be used by either if it's farmers, it's anybody within the animal production environment to actually enhance the product. And then this will efficiently translate the basic molecular, genetic, or cellular and clinical data into products that can be used at the clinical level and have health implications in animal healthcare. So here it shows the structure of animal translation bioinformatics in action. I'll go through the diagram as quick as possible. We have here different animal species that we might have. And then these animal species, we can get a lot of samples, a lot of whatever genetic material we do have. This material we can obtain the biosamples. These biosamples in form of blood, serum, saliva, urine, intestinal fluids, depending on whatever thing we use. These particular samples can be analyzed and produced in form of genomic data sequences, either in form of DNA, that is genomics, RNA, transcriptomics, proteins, or metabolites. Using predictive models of these, we can actually get different forms of these products using the predictive methods, including diagnostics, therapies, diagnosis and treatment assignments, and these can be directly applied into animal healthcare. But the question we also ask is, how beneficial is translation from bioinformatics? It's a very important question in the sense that if we have to actually translate bioinformatics, how to move this particular field, bioinformatics, the analysis of biological data, into insights, we have to integrate both experimental and clinical data streams into a more efficient workflow, effective management of resources and expenses. The reason we're putting that is no particular area has unlimited, for example, resources that are required. But if we efficiently manage whatever you're having and translate them, it will actually be a good way of actually moving data from lab to the bedside. And this will be a very important aspect because we shall be increasing or enabling that we have accessibility to data. We share the data and exchange our scientific ideas between the bench side and the clinical side. And it's a very important aspect in this sense. Another reason why it is beneficial to actually have translation bioinformatics is we can bridge the gap between the current reactive methods of health delivery and the intent of precision medicine. And in this particular area is drug development in animal healthcare. When you look at animal healthcare, most of the interventions occur late in the process, where treatment outcomes can less or reliably not effective. Generally, you'll find that by the time they intervene in a disease, anyway, in animal healthcare, it's really advanced stages. And therefore, we have to highlight a new way of precisely developing either preventive therapeutics or strategies that can actually be able to predict how this disease is progressing. And this is very important because it has been applied in human health or precision medicine, and the progress is really significant. Another important way of this is the methods that will be used to literally bridge this particular gap is data-driven disease classification, drug repositioning, as well as how do we identify disease biomarkers in animals, as they are done in humans, of course. And then one of the biggest of all is the creation of disease network models, each with significant impacts on drug development approaches in animal by combining the experimental data-driven approaches and probably computational prediction models. This is a very important aspect in bridging the gap between these two areas, the bench side and the bedside. And then, regardless of every single aspect that is positives of the translation bioinformatics, we do also have bottlenecks that exist in translation bioinformatics and with specific interest in sub-Saharan Africa. The biggest of all is the lack of investigators who are well versed in both the two domains, informatics and biomedical environment. And any effort to nurture these sets of competences within individuals will actually be a positive step towards having experts in this particular field. And then it will be a very good way of bringing in collaborations, that is interfield visibility. And it will also be a very important aspect to actually ensure that products, movement of information, I say data from the bench side to the clinical side, by combining the two areas. I'm not sure if we have individuals who can actually do that, the two domains efficiently well, but we would love to have a lot of collaboration that can actually bridge that gap between the biomedical domains and informatics domain. And this adoption of these will actually be a very good way of increasing application of bioinformatics in translation research in sub-Saharan Africa. And that will be across the fields of domain, whether it's from diagnosis, therapeutics, disease prediction, you name it, any of those. It will be a very important aspect. And what will be the way forward as having that we have particular bottlenecks? If we have to bridge that gap and have the proper expertise available, and it is possible if we construct very predictive multiscale network models of disease by you integrate the genotypes, the gene expression models, the clinical traits, and any other multiscale data to actually use it to infer different types of translation aspects of the research. And then this will be very important, especially when you're talking about the pathology of diseases or very specific treatment, therapeutic targets, or maybe diagnostics. Diagnostics is also an area. If you identify, if we combine the different expertise we have, we can actually merge from the biomedical field to the clinical side. we can identify the key drivers of pathology that can be very important or can play a particular role in identifying how do these diseases emerge in animals and how do they particularly spread to other places. And that will be a very an important addition to the arsenal that the researchers have in sub-Saharan Africa. In conclusion, we have many barriers in translation research. It is not one of the perfect areas that you can actually work in simply because of the different approaches that exist. But one particular thing that I am aware of, that if we connect the molecular world, the clinical world, and vice versa, it is undoubtedly that it will benefit both human and animal healthcare in the future, especially at the moment when we have a particular problem of animal to human disease transmission dynamics. And it's particularly important to have this featured across. And we are trying the best we can at the center to actually do that. With that said, I'll present a few of the selected projects or project highlights from the center that specifically work with translational bioinformatics. I'll be precisely a little bit quick to go through some featured animal research. There's development of antibody-based enzymes using a combination of molecular work and bioinformatics, finding out expression profiles for what do we call trypanosomiasis. That is the disease, quite a problem in sub-Saharan Africa. Another group of approaches that I used is modeling appropriate use of trypanosomides in trypanosomiasis. Then we have community-led data collection toolkits for the same trypanosomiasis. You also have some featured animal molecular epidemiology of diseases. This is from Rwanda. One of the students was from Rwanda, and he was doing research basically in animals and buffaloes to actually identify disease networks. And we also have some research from Mali, that is sub-Saharan Africa, on rabies, specifically how do different tests work and how do we manage to know which one will predict better a result. And then we have drug development for Cryptosporidium, diarrheal disease that affects disproportionately the African populace because of low resource settings. And then last but not least, we have analysis of different mitochondrial DNA in different populations of lions across the country. With that said, thank you very much for your time and attention. Unless there is any questions, concerns, feedback, that is all welcome. Thank you very much.
Thank you very much, Mr. Akarius. Before we pursue to questions, we'll pass to the second presentation, and then we'll come back to the questions later. So if you just dismount the slides, Mr. Akarius, thank you. In the meantime, while the second presenter mounting his slides, I will introduce our second speaker. So our second speaker is Dr. Daniel Janies, who is a distinguished professor of bioinformatics and genomics at the University of North Carolina. Dr. Janies received a Bachelor of Science degree from University of Michigan and a PhD in zoology from University of Florida. And he has worked from 1992 to 2002 at the American Museum of the Natural History in New York City, where he led a team that successfully built the world's largest computing cluster in 2001. Dr. Janies also has a ties with Ohio State University. He was a tenured faculty at the College of Medicine before joining the University of California. So his work involved empirical studies of organismal diversity, and he focused also on development of software. So one of such softwares is a SupraMap, which uses a genomics input with phylogeographic data to show evolutions of viral agents. Today, he will be talking us about understanding of the origin and the countermeasures to SARS-CoV and other human coronaviruses. So now the floor is to Dr. Janies.
Thank you, Zelalem. I'm going to talk about understanding the origins and countermeasures to SARS-CoV-2 and other human coronaviruses. Thanks for the kind introduction and the opportunity to address the audience. This is a paper we published in 2008 entitled Evolution of Genomes, Host Shifts, and Geographical Spread of SARS-CoV, now called SARS-CoV-1 by many people. And this is when I worked at Ohio State, and we made some news because we were among the first to identify bats as the fundamental source of coronaviruses. We did it from a bioinformatics perspective. All the data was publicly available. And on the right side of the tree, or right side of the slide, I'm showing a tree in which I've taken a phylogeny of viruses, SARS and animal coronaviruses that are not from beta coronavirus. And what really inspired me to do this is, I don't think she's on the line, but Linda Safe is a senior coronavirologist at Ohio State, and she had come back from a meeting of the World Health Organization in response to SARS-1. And she showed me some of the slide decks that the phylogeneticists in China, led by Yi Guan, had created. And this was before SARS in bats was widely recognized. And what they had done is they had some interesting data. They had what I'm depicting here in yellow, a lot of human SARS cases, viruses extracted from humans. And then they also, what I'm depicting here in purple, they had SARS extracted from small carnivores in markets in Shenzhen, China, for example. And what they did was just graphically forced the sequences from carnivores to the bottom of the tree and use that as an argument to say, you see, carnivores are the source of SARS, we got to kill all the civets. I imagine they were under intense pressure to come up with some sort of solution. So there were large civet culls in China, and now we all know it didn't do any good because there may have been something going on locally in the markets in Shenzhen, exchange of virus between civets and their handlers, and it went both ways. As you can see, that purple line comes out of a yellow line, so it went, I don't know if you can see my cursor here, but this is the past, this is the present. SARS went from yellow humans to carnivores, spread among carnivores, and then back to humans. So this was a very localized event in Shenzhen. But fundamentally, what we're after here is the root of this tree. And the way we discovered that bats were the fundamental source of SARS-like viruses is we took a lot, and this was really inspired by Linda, took a lot of these animal coronaviruses, okay? And we knew they weren't SARS, we knew they were something else, so they must share a deeper common ancestor than any of the SARS or its recent ancestors do. And so by doing that, we can find this deeper point, and that allows us to put a baseline in the field of phylogenetics. We call that an outgroup. We use an outgroup method to root the tree. We don't force anything, we discover the root. And once we discovered the root, by comparing to non-SARS coronaviruses, we could read the history. And here we read black bats to yellow to humans to carnivores, and then also spreading throughout the human population, even went into pigs at one point. So we were among the first, in summary, to discover the fundamental origins of coronaviruses in bats. So I have some goals for today. I want to understand zoonosis to develop strategies to defer future emergences of viruses from animals. I want to understand possible animal models for the study of coronaviruses. I want to understand when BSL-2, standard wet lab viruses, can be used as models for BSL-3 high security viruses. I want to understand how viruses become resistant to therapeutics. And I want to understand the history and movement of coronaviruses. These are all big questions. I'm probably just scratching the surface of each. To help achieve these goals, our contributions are developing and sharing gene annotation tools and phylogenetic visualization tools, We have created these in the past. One of them was called Supermap. Now we're focused on a tool called FlaviWeb, which is publicly available and published, and StrainHub.io, also published in the peer review literature. We are using methods to use as much of the genome as we can reliably find as orthologous among viral families to increase the explanatory power of our phylogenetic analysis. So we try using especially annotation tools like FlaviWeb, which we've adapted to coronaviruses, to find areas of like genes across different coronaviruses. And I'll explain that a little bit later. But fundamentally, we use phylogenetic trees. Phylogenetics is how we understand relationships of our strains and read host shifts. And you may, if anybody who works in metagenomics has heard of the term OTU, it's a fancy acronym for operational taxonomic units. In human terms, it really just means the tips of the tree, the A, B, C, D, and E are the observations. They're actual viruses or bacteria or animals or samples. This is the biology. Down here, these HTUs, these nodes, so to speak, in this graph, these are the hypothetical taxonomic units. These are the mathematical constructs that we use to join the observations and hypotheses of similarity, typically through a computational search for shared mutations. And as we add OTUs, this tree gets bigger and bigger, and there's more possible shapes of this tree, each one representing a hypothesis. And that's why we use high performance computing, because this problem explodes combinatorially and in various levels, not just searching the trees, but also finding which, bases in the DNA or RNA, or which amino acid in the protein corresponds to which in other organisms. But one of the most important things that's it's really become a lost art is how to actually read these trees. And most simply, we're looking as the tree not only as a hypothesis of interrelationships, but we want to read it. We want to derive information from it. And we can derive all kinds of information. It's really only up to the creativity of the user. So what we do is we kind of paint the tree or layer other information on it, such as anatomy, or we look at the actual mutations upon which it was built. Place, we can look at shifts in origin of place of a virus, could go from Charlotte to Columbus, for example, or hosts could go from bats to humans, for example. So this is the past on the left. I've turned the tree on its side here. And this is the present on the right. And so we read the tree, black being sort of the ancestral state. And then in this branch, we had a shift from black to red. Let's say that's, Charlotte to Columbus. And in this branch, we have a shift from black to blue. Let's say that's Charlotte to Raleigh or something like that, okay? And these black and red and blues can be anything you want. It can be bone long, bone short, bat host, human host. Could be position 600 has an A in one lineage, and position 600 has a C in another lineage in DNA. And so we call this, the fancy word for this is apomorphy list. It just means change, a different shape, apomorphy, just changeless. So we tally up all these changes, okay? And we'll get to how we use that in the future. But here, back to our original example, adding some more modern history. I talked about SARS-CoV-1 here. Now we all are acutely aware of SARS-CoV-2. And I think with some hope, I put 2020 on here, but unfortunately that'll have to be revised to 2021 soon, and perhaps further years. And MERS, Middle Eastern Respiratory Syndrome. And this is actually also an error. MERS still circulated until about 2018. But the point here is this is the past. Fundamental origins of these human important coronaviruses in bats, here illustrated in blue. And then if you follow the color change, it goes blue to yellow, okay? So you go directly from bats to humans in all these cases. So bats are not only the fundamental source of coronaviruses that cause SARS-CoV-1, they also are the fundamental source of SARS-CoV-2 and MERS. MERS is a little more complicated because it does require a reservoir host. The Camel's Artiodactyla uses a different receptor system, and it's almost as if MERS infected the herders and the camels at the same time. It's just circulating among them. So it's a bit of a different situation. But with the SARS-CoV-1 and CoV-2, we're strong believers in what we can reconstruct from the tree, direct infection of bats. And what I want to point out here is all these dotted black lines. These are not important to, with the exception of camels, these are not important to the human infection of SARS-CoV-1 or CoV-2. So hedgehogs, scaly anteaters or pangolins, birds, small carnivores, pigs, different kinds of small carnivores, not just civets, but raccoon dogs. These are all just spillover events into husbanded animals, and they weren't reservoirs for SARS-1 or 2. This is pretty complicated. This is what Dennis Jacob Machado is doing in our lab, but it's an entire workflow. He's looking at 2,000 coronaviruses of all sorts, alphas, betas, deltas, and gammas. He's creating an annotation pipeline to try to find these orthologous regions. We're using almost the whole genome. He's found it useful to focus on ORF1A, 1B, S, E, M, and N as regions he can reliably identify as common among these diverse coronaviruses. And here we're studying different hosts, okay? So for example, these deltas and gammas are popular in birds, and here's the carnivores in these purple triangles, and humans are these red plus signs, okay? And you see human coronaviruses have occurred many times, and I'll talk about that. And then we can count up the sort of source animals. This is the same chart twice. It's just there's some error bars, so to speak, min max changes of what we can count out of this large tree of coronaviruses. And you can see Artiodactyla contributed human coronaviruses many times, and that's what I illustrated earlier, the camel-human exchange in MERS. But Chiroptera, here, this green sort of checkbox contributed humans coronaviruses at least 3 times. We reconstruct none from Eulyptopora, the hedgehogs, for example. And then humans have given coronaviruses a few times to carnivores, that's probably SARS-1, and a few times to Artiodactyls, the camels, and so forth. And here's SARS-CoV-2. And importantly, I want to show here, these are the Phylodota, or the Manus javanica, the scaly anteater or pangolin. That's a completely different transmission from the ancestral bat sequences or bat hosted viruses to pangolins. And then the bat hosted virus, completely independent of that shift, contributed SARS-CoV-2 to the human population. And then there's very little resolution. You don't see any branches from this, because basically SARS-CoV-2 exploded in humans with almost no evolution. And so we haven't created a map of SARS-CoV-2 because you need resolution in the tree. If we add many outgroups, we can get some resolution, and I'll show you a little bit of that later. But all these graphics you see in Yahoo News and so forth, they're not built on mutations. They're built on something else, or an improper interpretation of how phylogenetics actually works. And so here's just a blow up of that host shift I've been talking about. So we want to use common features among SARS-CoV-2, which requires BSL-3, which is a rare resource, and other coronaviruses that you can buy from lab suppliers, OC43 and 229E, which cause much less severe disease in humans, and they only require BSL-2 support. And this comparison prevents some advantages for drug repurposing. We can open up the whole biological, research enterprise to work on coronaviruses as a surge to respond to them if we can open up all the BSL-2 labs. So BSL-2 strains can serve well for assay development, such as antibody detection, animal models. Drug targets are found in highly conserved regions, and this makes the therapeutic more applicable across coronaviruses. That's an open research question, but for example, remdesivir, which you may have heard in the news, is widely applicable across coronaviruses. Well, there's some disadvantages to this sort of broad-based approach of comparing across coronaviruses in terms of drug development. Only parts of the genome of SARS-CoV-2 shares conservation with other coronaviruses. We want to know which those are. Logically, you would think conserved loci are unlikely to be deleted, but it does occur. The E gene can be deleted. There's E gene deficient lineages of coronaviruses that survive in labs. And conserved loci can vary in point mutations that confer resistance. It's not just present in the whole gene, but some of these small molecule drugs, such as remdesivir, as applied to coronaviruses, or Tamiflu as applied to influenza viruses, are susceptible to a single point mutation that can confer resistance. So here's a chart made by Dennis again, and in the first column we have the genes, and black are ones that we would not suggest for study across coronaviruses. We cannot, for example, find commonality in the non-structural protein or the hemagglutinin esterase, but the rows that are in red, we can find commonality among 229E, OC43, and SARS-CoV-2. So ORF1AB, the spike protein, the envelope, the membrane, and the nucleocapsid, that's what we're using to build our phylogenies. And what we could suggest people who want to use BSL-2 labs would focus drug targets on, because they're likely to be applicable to SARS-CoV-2 in translation in BSL-3. So here in the United States, our Food and Drug Administration has provided an emergency use authorization for Gilead to use remdesivir in treatment of SARS-CoV-2 infections. This was done in May 2020 and renewed in August 2020. The president was treated, among many drugs, I'm sure, with remdesivir. If you go to the hospital here in North Carolina, my physician friends said it's available. It's apparently rare though, so we're lucky to have it. So remdesivir stops termination of the RNA synthesis as the coronavirus hijacks your cells. It's a therapeutic, it's not a cure. It lessens the severity of the disease, much like Tamiflu lessens the severity of an influenza infection. The drug target is the open reading frame 1B. The open reading frame 1B of the SARS-CoV-2 genome encodes the RNA-dependent RNA polymerase, which is the target of remdesivir, a drug that has shown effectiveness against many coronaviruses. And there's an important paper by Brown et al. In antiviral research illustrating this. And this is work done, I would assume, in 2018 or previously, published in 2019, so it's pre-SARS-CoV-2. So here's their tree from Brown on the left side of the diagram. And they're using colors and they've got animals, so it really appealed to me. You can see in blue that these remdesivir was largely developed against these bat viruses. This is very, very clever of them. In MERS, they color code it a little bit in black, a little bit in red, because it's a camel human virus. I thought that was very clever. And then you have SARS-1 here. They depict as a human virus and so forth. But the most important thing is that these sequences of these viruses share an F at this position, position 476 in the amino acid sequences of the ORF1AB polyprotein. And that that's the wild type, let's say, that makes them, all these viruses can be stopped by remdesivir. They're susceptible. And they occur across the spectrum of coronaviruses. However, there's some of these avian coronaviruses, these deltas, that bear an L at this position, and that renders remdesivir, well, they are resistant to remdesivir. So I checked the databases for SARS-CoV-2. I checked these positions. Here's the F in the ORF1B polyprotein. So most of SARS-CoV-2 remains, thank goodness, susceptible to remdesivir. It'll work. SARS-CoV-2, unlike influenza viruses, does not evolve extremely fast. Remdesivir should be available to us for a while, but this is definitely something to watch. There are other positions, not this tree, but the second to the last V here, in some cases confers resistance to remdesivir as well. So we had done similar work on drug resistance, putting it in a phylogenetic and geospatial context before, and this is done in SupraMap. Basically, we take this phylogenetic tree, this branching diagram, and force it into the earth, contort it and project it much like an image projected on a screen into the earth. So at the tips of the tree are at their geographic positions in the earth, and then you've got this sort of scaffold going up in altitude, representing the HTUs, so to speak, all the way up to the root of the tree. But most importantly, what we've done here is we've colored the lines based just on one position. Orange lines represent lineages with the genotype 31N of the MT protein, and this is H5N1 influenza. And this mutation confers resistance to adamantane derivatives, another kind of drug. The white lines represent lineages containing the genotype 31S at this position that are susceptible to adamantane derivatives. So you can see here in the ancient history of the H5N1 that emerged from central China, it was susceptible to adamantanes. But importantly, it's gained resistance multiple independent times throughout its history and its geography. It came over to Indonesia. You can see this large arcing white line. Came to Java as susceptible, but gained in Sulawesi resistance, spread to Sumatra and other parts of the archipelago as resistant. Independent of all that, you can see these sort of lineages popping out. When you're close to the Earth, you're more recent, popping out of central China and spreading throughout Western China. And then some older lineages popping out of China, conferring resistant lineages in Vietnam and Thailand, and then spreading subsequently to Malaysia. So the point being here is we see lightning striking multiple times, and we use another calculation to actually look for what's called a skewed ratio of non-synonymous to synonymous mutations. And this was well beyond Darwinian or neutral expectations, and therefore we could say that there's Darwinian selection in this on this position, on this change. And that's probably by overuse of this drug. Even, and apparently reports suggest it was even used in agricultural purposes. Remdesivir being rare, hard to manufacture, expensive, extremely valuable in therapies now, will probably not be used on the farm. But you know, the biomechanics and the evolutionary pressures could be the same. Let's say we made remdesivir widely available and cheap. So we looked at remdesivir resistant lineages above and beyond what Brown did, and we see many viruses. These are mostly Delta coronaviruses in birds, but also some other kinds of organisms, swine, for example. And these are quite close to home. Here's one out of Ohio. And here's some out of, these are porcine coronaviruses out of Illinois as well. And one of the things, this is not by any means anything we've demonstrated, but viruses evolve not only by mutating, but by exchanging genes through recombination. And here's just an illustration of how that happens, how we would discover it in a tree. This lineage over here would contribute genes in a horizontal way rather than an ancestor-descendant change. And this is a big avenue for phylogenetics, because the software that was developed to understand this is old, runs on Windows computers, and only can examine a few viruses. It's a very complex problem that is sort of different from the way we think of things. We always think of things changing from root to tips, and this horizontal events have been poorly examined, and so there's avenues for improvement there. And here's the application. We would look at whether deltas are exchanging with beta coronaviruses, including SARS. One of the things we've done most recently in geography is a new way of viewing this information rather than just forcing the phylogenetic tree into a geographic map. We're trying to extract the information from the tree and explain it in a much clearer way. So we've created a tool called Strain Hub. We've published that in Bioinformatics earlier this year. What Strain Hub does, it takes not the tree, but reads information from the tree, much like I showed you in that black to blue, black to red diagram on slide 2. And it makes a network out of that information, okay? And we can derive a lot of information from that network. Here we have a Strain Hub graph of the spread of SARS-CoV-2 in China, 2019-2020, rooted deeply on a SARS coronavirus out of bats. So SARS-1 and SARS-2 share deep common ancestry. And again, killing the civets didn't do any good. It was SARS-like viruses have been circulating in China for decades, and they just come out into the human population every so often. So this is one of these, deep ancestors, or one of these viruses that shares deep ancestry with SARS-1 and 2, and that having that in the mix allows us to find some resolution in the tree as well. This is something we're working on. But we took that tree and we made a Strain Hub map. We took all the transmission events out of it, all the changes in places. For example, you see these arrows, Hubei to Beijing, or Hubei to Henan, Hubei to Shanghai, and so forth. And they have polarity. The arrows represent the polarities. The thickness that they have frequency. The thickness of the arrows represent how frequent they are. So you see very frequent exchange from Hubei to Guangdong and so forth. They can go back and forth, Hubei to Yunnan, Yunnan to Hubei, and so forth. The most important thing here is we can go beyond just visualizing it. We can analyze this network much like you would analyze a social network. In a social network, there are influencers who have more connections. And one of the ways we measure those connections is centrality metrics. In this case, it's one we come up with in my lab called the source hub ratio. And it's basically a 1 to 0 metric that says if it scores 1, it's a source for viruses, or a sink near 0, it does not spread viruses. And this could be this diagram could be for hosts or places or even people if we had the ethical approval to do that. So we've not only depicted the source hub ratio in the size of the circle, but we also have this diagram here. So we can look up Hubei is very central to this network. Hubei is where Wuhan is. You can see its score is very high in this source hub ratio. So for SARS-CoV-2 in China, Hubei is very important, but also Beijing was very important as well, has very high centrality. Oddly, Shanghai, arguably an incredibly important city in China, doesn't score very high. And I have a boyhood friend in China, and I visited him during H1N1, and just the difference in how the world reacted to H1N1 and SARS coronavirus 2 is striking. We had tools to deal with H1N1. We knew how to make vaccines. We had Tamiflu, but it was just 11 years ago, and it's such a different time. And I took this picture in the middle of the H1N1 pandemic in Shanghai, and everybody's going around about their business. He took a picture on one of the train lines. It looks almost the same one. And my friend Tom, in February 2020, it was dead as a doornail. Okay, and we all know about the lockdowns, which have been variably applied around the world. And we think this source hub ratio, which depicts very low transmission in Shanghai, is reflective of the severe lockdown in China, especially in Shanghai. So tree time is another tool out of Nihir's lab that we've been looking at. This is really Dennis's work. I'm more of more interested in character change than molecular evolution, although he's convincing me to get interested in molecular evolution. And basically what happens here is you stretch, we're doing more contorting and stretching of the tree. We stretch the tree such that the branches are sort of calibrated on this graph of time. And long branches mean there's a lot of evolution. Short branches mean there's little evolution. Okay, and it's basically a regression to fit this. We're lucky in viruses. It's not like working on dinosaurs or animals where we don't know the date. Viruses come with timestamps, so this is much easier than it should be. And one of the things we've done is we wanted to see if we could reconstruct the date of emergence of these 9 important lineages for humans, for human medicine, of coronaviruses. And he's done quite a good job of it. These dates calculated in tree time correspond very well to news reports. For example, SARS-CoV-2, here he's got it on Christmas Eve. It probably emerged before that. We looked at when it emerged out of bats, and it was about 8 years ago. And we have their mutational rates. You can see, NL63 evolves relatively fast. OC43 very slowly, SARS-CoV-2 somewhere in the middle. And so these dates are pretty accurate. I'm just astonished how accurate they are. They're a little late maybe, but again, we don't get the first viruses. We have to see it emerge in people first and decide we want to sequence it. And we're starting to go to the literature and see when the first papers about that virus were published, whether they be electron micrographs or cultures or pre-molecular techniques, and then also the first sequences when they were established, and try to get an idea of sort of the library science around these dates too. But I'm just astonished how well this has worked. And one of the points I want to make from this in conclusion is that this is just Excel math. It's not very precise, but the months between emergences. So if we compare the emergence date for NL63 to 229E, we got about 148 months or 12 years. But then we started to get, hit with these things more often, or we were aware of them. I think the senior coronavirologist could really illustrate what was going on in the 80s for us. That would be very interesting to me. So we started to see more of these things happening, OC43, 4408, HKU1. And what's interesting about this column here, as you can see, it's not really periodicity. These things don't come in a cycle like cicadas emerging out of the forest, but they come in handfuls of years. And one could say, if you wanted to make a soundbite, they come every 10, 12 years, much like a US census. Or you could say they come every 3, 4 years, 7, 8 years, like Olympic Games. So my point being here is coronavirus zoonosis from animals to humans is not rare. We used to think of these things as rare events. They're not rare. The data is quite clear. The devastation they can do is quite apparent. So we should really start to take this field seriously and ask ourselves what is next, and go from retrodiction to some forecasting to the extent we can. Thanks for your kind attention. I'd like to acknowledge all the lab members who did most of the work. Dennis did the bulk of it. Colby and I are working on Strain Hub. Sayal and Rachel are extremely valuable for data collection. And I'd like to acknowledge the organizers and audience, the OSU Global One Health Initiative. Thank you very much. I was just thinking about it. It was just a year ago we were in Quebec, and I hope the world gets normal and we can do that again one day. I'd like to thank some of our benefactors, the Belk family and UNC Charlotte. So thank you very much.
Thank you very much, Dr. Janies. For participants, there are 2 ways you can pose your questions. One, you can just put them in the Q&A box, or you can raise your hands so that Dr. Binkley can see, so that she can unmute you to talk. So with that said, I will progress to the Q&A. So let me give the first opportunity to Dr. Binkley. She has a question. Maybe she will ask it herself.
Thank you, Zelalem, and thank you both for such wonderful, highly significant presentations. My question is actually for Acadius. So there's a general movement towards whole genome sequencing for identification and phylogenetic analysis of circulating viral and bacterial variants within a given area. However, sequencing of complete genomes produces a mass amount of data that needs to be analyzed, which can slow down surveillance systems if there's limited bioinformatic capacity. So do you see whole genome sequencing as a sustainable standard for disease surveillance systems in sub-Saharan Africa in the near future? And if not, what do you think will be required to reach this standard?
Thank you very much for the question, Dr. Binkley. If I have to be honest enough, I should say that the bioinformatics capacity in sub-Saharan Africa is still quite in edges behind a little bit, to be very honest, because when you talk about the sequencing is done, most of the sequencing is done outside. You actually send your samples for sequencing to actually have them. So that is already bottleneck. The access to, even if you're talking about the whole genome sequencing, just that alone is a bottleneck already, because how do you have to send samples for sequencing? And most of the time, people just sequence small sections of the data we're having. That is what basically we actually use. Rarely use, technically, if you say the whole massive data sets that can be analyzed. We use the available data sets that are available to actually do anything that we want to do. So even before you move to the next stage of saying that it is quite restricted in surveillance, even data itself, actually, unless the public libraries, or what do I say, something like NCBI, but locally the data we have, we use maybe this particular section of data or a particular, it's more a targeted approach. So that is what we basically use to actually do any form of translational bioinformatics or any form of bioinformatics, unless I didn't get your question very well.
No, thank you. Excellent. So really, partial genome sequences, working to maximize those might be something that would be a little more beneficial at the time.
Pardon?
So using partial genome sequences, that may be something that's still sort of learning how to maybe maximize those efforts would be a better investment of resources than jumping to full genomes, perhaps?
Yeah, that is exactly what I recommend, simply because most of the time people do targeted research because of those constraints. So it's a very particular form of designed project. So rarely do you have, unless you have collaboration. One of the highlights that I produced was actually getting experts in both areas, where you have somebody who can actually do proper informatics. Merging with the biomedical data is a quite challenge that is actually understandable. But notwithstanding that particular fact, is the available data that is locally generated is quite targeted. You look at the kind of the projects that have been done, is limited to a particular area or a particular targeted research. So that is what we just simply use for any of the things that we can actually try and work out. For the whole genome sequences, the most cases, the ones that you get will be collaborative research from different organizations. And most of the time, the research has a timeline, probably 2 years, after which the data is just deposited. And thereafter, any other effort is actually not possible because it has to be funded, which is not funded. And how much you will try to actually have a collaboration, you require the funding to actually proceed from, if you have to analyze the large data set. Another important drawback of the same is, take an example like, for example, the analyzing something like a whole transcript from either humans or whatever it is, a whole bit of it. The computing power might not be available to actually run, say something like a network analysis simulation or something of that sort. And that one is already a limitation. So what you'll focus on, can we actually work with a smaller section that we can actually manage within our resource limits and use that to translate into an actual useful product that can be used on the ground then, or by, do I say an output of the same?
Excellent. Thank you so much.
Okay, thank you.
Please, if you have a question, please drop it on a Q&A box to all participants. But in the meantime, I can throw one question to Dr. Janies, myself, and then we go to and fro as needed. We will not going to be following any patterns. As far as the question come, we'll give them the opportunity in order. So Dr. Janies, thank you very much for that great presentation again. My question is, you mentioned about the SARS-1, which arose in some 20 years ago, I guess. And then you talked about a little about the civet cats, the intermediate hosts. That was interesting to me because we still don't know the intermediate host for SARS-2, yeah? So can you comment on how much not knowing the intermediate host could affect this pandemic mitigation efforts? Because you emphasized, like, you know, the presence of this intermediate host probably could be a potential continuous spillover source to humans, or, you know, if we have a vaccine, how we are going to mitigate it, things like that. So how the intermediate host factors in the whole pandemic mitigation efforts. I will have another question, but if other people are not asking, I will come back again.
Oh, I would argue there are no intermediate hosts. The civets were a sideshow. Okay. So I know if you look in Wikipedia or even the World Health Organization, that's a common misperception there. And there's been a lot of bat denial, but we showed in SARS-1 and we showed in SARS-2, at least with the data that we have at hand, bats are the fundamental and proximal hosts of SARS-1 and 2 in humans. There's no need nor evidence for intermediate hosts. The only intermediate host that I would say has any legitimacy is the camels in MERS, but that's a different virus, uses different receptor biology and had a different history.
Okay. So if I'm not clear, what was the premise? I don't know. I'm not clear, very sure what the premise was to propose intermediate hosts for SARS-1 and SARS-2.
I don't know. I mean, I think people have always thought that it was necessary. It was that there's this notion of a reservoir where the virus has to go hide. You know, we can eradicate it, and we treat ourselves, right? We can eradicate it in humans, but these things reemerge, right? And dengue, for example, has a reservoir thought to be in non-human primates, and but it occurs in a lot of small mammals as well. And so that, I think that's just in the viral psyche that there's a reservoir necessary, and bats didn't enter into that equation. Bats spread a lot of virus, sometimes through a reservoir. In the case of SARS-1 and 2, no reservoir required.
Okay. Maybe in continuation with that, because I saw one of your slides, you do this comparative genomics using the different ORFs. And then SARS-CoV-2 sits next to the rat G13, which we know is the most, closely related bat virus we have. Yeah. And I think with this intermediate host theory, and as well as with, some other most people have been, tied with the furin cleavage sites where it appears. So I think that precipitated with the questions of this intermediate host. Yeah, how do we explain the appearance of this furin cleavage site in SARS-CoV-2 if we don't put intermediate host into the picture?
Well, that's one feature. And so that's why we're using the whole genome. We're using many features. So yeah, people have argued that the pangolin biochemistry looks closer to the human SARS biochemistry. But the evolutionary history of the whole genome is I would say more evidence and therefore a stronger argument. And the evolutionary history of the whole genome points to bats, as did SARS-1.
All right, thank you very much. Thank you very much. Any other questions? I don't see any questions coming as of yet in the Q&A box. Maybe I will throw one question to Mr. Akades as well. Maybe it's an overlapping question. It can be answered by both of you, but just to start with Mr. Akades, you've talked about like the lack of investigators in sub-Saharan Africa, especially a constraint when it comes to bioinformatics and using these data science approaches for, you know, investigative research or clinical applications. So how do you see like the training goes? Because you mentioned that there are two domains, the one with the informatics or the bioinformatics domain, those people who are trained in that. There are people who have also a bench experience. So in bioinformatics, we need these two things to tie together. And so how do you see the current curriculums or university teaching methodologies addressing that, and what kind of you know, things into the future could mitigate, you know, producing competent professionals that have a computational as well as experimental skills? And maybe I'll start with Mr. Akades.
Thank you very much, Zelalem. Just a point of correction is not that there is lack of experts, there is lack of a proper experts who can actually pick up. You can be an expert in both areas, yes, but it's proper if somebody has, say, the informatics side properly done, and then somebody does the biomedical side, and then there is a merger. But rarely does that happen, because whatever has happened is to merge the two disciplines and just have an overlay. You have some knowledge, biomedical data, and you have some knowledge in informatics, and there you go. Then we have the bioinformatics. That is basically what we're having. But we'll appreciate if there was a lot of collaboration between different places where you have, for example, somebody with proper knowledge about computing, crossing over to actually come and collaborate with somebody exactly with deep bench knowledge. And then these will actually, when I probably am from the computing side, if I raise a question, or from the biology side, if somebody raises the question, it can be actually be put up into the informatics side. Take an example, I have something to actually do analysis. Can somebody pick up and do an algorithm that can actually help me do that? Or for example, I have some algorithm I feel can work for the logical side to actually bring out probably an output that is very important. Now, how do I come and collaborate to get that? Because that will be the best line. Bioinformatics has been around for quite more than probably 2 decades. Relatively, that is not very old. But I think the approach we basically use is a particular problem with I will not call it a problem per se, a particular approach where you were taught a section of the work in computing, and you taught a section of the work in biomedical informatics, biomedical data training, and that actually produces an individual who is neither both, who is neither of the two. So I'll appreciate if there is more hands-on training later on, is where you have repeated collaboration, what would do. Because when I look at the work Dr. James is doing, the work for phylogeny, I look at it and say it's very focused and very particular to a particular area, in the sense that they have tools specifically for that particular work. But when we come across these other side, it's a little bit different, because we have to get, you have to leave your lab, come sit down and write the code to ensure it's running. That's a particular problem. And that one actually means that most of the work that we'll have done, the other side, probably for the expertise you'll have built up for the lab to actually be very precise, will actually be, the tension goes both sides. That's one particular problem. And that's the reason why the translational part is a problem, because we have a jack of quite a number of trades and a master of none. And that's why we're calling for more collaborations. Can we have collaboration from A to B to C? And so that we have this school, how much us as a center have that particular collaboration, we still struggle post-class to actually have it work up. But it is not all in vain, because we're really building up some collaboration. As you can see, there is something known as Bionet that actually is doing a lot of training. And that's what we really want, so that you can actually build the capacity building for both sides. An individual with the informatics side can actually get a lot of data over training over time. It's more professional development over time. And not that there isn't, but it's not that the expertise we have not sufficient enough to actually do the work, to actually create the translational aspect properly. Thank you.
Thank you, Mr. Akade. So I'll forward the same question to Dr. Janies as well. If you could comment, especially from the US perspective, and how the question is tackled in US.
In terms of cross-training?
Yes, cross-training, because for example, in the US, I see a lot of graduate students are taking a lot of computational courses, trying to compensate, from the skills which they have from bench sides or the laboratory trainings with some computational skills. So it seems that the era requires like a person who's trained to conduct or somehow have the biological side as well as the computational sides. So
So we've addressed this head on. We are in a college of computing and informatics. I trained as a biologist, and I picked up computing along the way. But now, we're training people straight out of essentially, undergraduate in both sides. So if you are primarily, have aptitude in biology, if you're going to work in bioinformatics, you're going to have to learn the computation. If you're primarily a computer scientist in your training, you're going to have to learn the biology. Some people argue one direction is easier than the other. I'm not sure I subscribe to that. I think humans are adaptable, no matter what they put their minds to. And even our colleagues, we do have a College of Literature, Science, and the Arts. Even our colleagues in biology over there are starting to join forces with us. If you're not doing bioinformatics, it's very difficult to advance in biology today. It's very difficult in the US context to advance without some omics approaches in your work.
Thank you very much, Dr. Janies. I think I see Aseta raising her hands. Laura, if you could unmute Aseta to ask a question, we may have a time for a quick question.
Aseta,
you can go
ahead.
Aseta. Aseta, do you hear us? Yep, you can go ahead and ask the question. Okay, maybe
I can ask a question. One question, a little bit of a clarification from Dr. Janies on the presentation.
Sure.
There is something about repurposing. You talked about something on repurposing along through the presentation. I don't know if you've looked at anything about which strategies are there for repurposing and which ones will possibly be of use.
Yeah, not me personally, but we're aware of a drug repurposing group at University of Michigan who have a really nice system. They are infecting in cell culture, a variety of cells, SARS-CoV-2 in their BSL-3, and they have high throughput screening methods. And the way they've enabled those through computation is through computer vision to characterize the infection and then the result of the drug on the infection by processing the images using machine learning. And that's a very impressive group. There's other groups around the country doing that, but those are the ones we're most familiar with. And they've put up some preprints of potential therapeutic agents that are already approved and could be brought to bear on SARS-CoV-2.
Thank you.
Thank you.
Okay, it's 9:52. We have a few minutes. Maybe I will just throw the question, one question. If you volunteer any one of you to answer, I would be very happy. It's related with SARS-CoV-2 again. Maybe I'll start with Dr. Janies. Do you have any opinion on this spike protein D6140G mutations, how it is circulating? I know you said like you're not interested in molecular evolution, more of
I am
rather yeah. So if you have any opinion on the
that's the sort of the population of SARS shifted over to this mutation, and what's the phenotype of it? It makes the infection more severe. Yeah, I believe it. I mean,
okay.
There are only like 8 mutations. That's one that has a clear history. The problem for me is it's very hard to build trees on 8 mutations. I believe it. Oh, what it means is not clear to me yet. So, is it just a feature of the population genetics of the virus, or is it an adaptation? I don't know yet.
All right.
Maybe a question to pass by to Dr. Binkley and Zelalem. Knowing that you've seen what we're actually doing, what particular collaborations do we have, or how can you, from the side, do you have any similar thing like translation bioinformatics that is taking place that probably we can collaborate and have, even if it's virtual, for more of the training part? In that sense, like virtual, maybe we have classes or something that can actually improve that particular, because that is an important aspect of way forward for most of from the sub-Saharan, from this side's perspective.
I completely agree with you, Mr. Akari. So, as you know, Global One Health Initiative has a very good tie with Eastern Africa, and we collaborate, especially with genomics and bioinformatics specific questions you mentioned. You know that we are currently actually collaborating with the University of Nairobi and working on a data science proposals. We are also doing the summer institutes, which we're supposed to undertake on ground trainings and also some data analysis training, which we did virtually with, in collaboration with Charles Zuckerberg BioHub. There are several collaborations that goes from Ohio State to University of Nairobi, and also university in Tanzania and Ethiopia as well. I do believe we have to strengthen that, and we have to also, make it resilient when we have a similar situation that we have now with SARS-CoV-2 when travels are restricted. So we opt into a virtual mode and try to conduct these trainings in a virtual mode. We try to introduce these new technologies. Also, we have a plan bringing some postdocs and also going there and giving training on new technologies like nanopore, long-read sequencing technology that can probably have a very big potential future in Africa. So there are works that has been done. There is a works in plan. That's what I can say from my side. When I look at the time, I think it's 9:56 a.m. So it's a time to wrap up, I guess. So I would like to thank both of our presenters very much for such a nice and interactive sessions. I hope I will see you with another format or in another way in the near future. Saying that, I will pass the podium again to Dr. Binkley. Thank you.
Hi, thank you so much. We do have, it looks like, a quick question from Aseta, if you're able to speak. Can you hear us? Looks like Aseta dropped off. Okay, well, with that, thank you so much. And again, to follow up with what Zelalem was saying, the GOHi does host the Global One Health Summer Institute every summer, and bioinformatics is certainly a part of that. So yes, happy to have anyone join. So thank you so much for joining us today, and thank you to our wonderful speakers. We look forward to seeing you in November. And with that, we'll go ahead and close. Thank you. Have a good day.
Thanks very much.
Okay.
Thank you all. Thank you.